experiment is on the effects of endosulfan exposure on brain neurotransmitter levels in African clawed frogs. Understanding the impact of pesticides in amphibians is a growing concern. Among pesticides, endosulfan belongs to one of the most dangerous sources because of its wide use and known effects on organisms. In this research, tadpoles of the African clawed frog were submitted to 4 treatments during 27 days.
1/3 of amphibians are threatened with extinction. One suggested cause of this decline is pollution by agricultural contaminants, which run into the waterways. Pesticides are applied in the spring, which is when amphibians reproduce, leaving larvae vulnerable. Endosulfan is a high toxic insecticide that affects the nervous system of organisms. It is known to mess with acetylcholine, serotonin, and dopamine pathways, as well as cause neurological disorders, hyperactivity, convulsions, paralysis, and even memory problems. The objective of this experiment was to investigate how low concentrations of endosulfan induce different neurological and behavioral traits.
African clawed frog tadpoles were exposed to 4 different treatments: a control, an ethanol control, a 0.1, and a 1 micrograms per liter nominal concentration of endosulfan diluted into ethanol. There were 30 tadpoles per treatment, and each tadpole was exposed for 27 days. The ethanol control was used because endosulfan was diluted in ethanol. After 27 days, tadpoles were snap frozen on ice, and the tails and the brains were dissected to measure for gene expression. There was a physiological analysis on serotonin, dopamine, and GABA. There was a molecular analysis performed on brain samples. ELISA tests measured concentrations of serotonin, dopamine, and GABA. There were acetylcholine assays which monitored acetylcholine activity in the tails. PCR analysis was used to analyze DNA, and behavior variables were monitored. There were visual determination to track feeding and video tracking to watch locomotion patterns. The results for neurotoxicity markers showed that there was a significant increase of serotonin in both endosulfan treatments compared to the normal control. However, there was only a significant difference in the high endosulfan treatment compared to the ethanol control. Dopamine and GABA increased significantly between low endosulfan and the ethanol control, but not between the high endosulfan and the normal control. Acetylcholine activities in tail muscles varied. Although there was a slight general effect, there was no significant effect in acetylcholine receptors. The results for the gene expression showed that there was no significant changes in gene coding for brain neurotransmitter receptors or for metabolic enzymes. As shown in the image by the arrow, only the gene coding for GABA transporter 1 was modulated according to both endosulfan treatments compared to both the normal control and the ethanol control. The growth and behavior results showed that the effect of treatments on body size and weight was only marginally significant. There was no significant difference between the ethanol control and both endosulfan treatments, but there was a significant difference between the normal control and high endosulfan treatment for body size and weight. Feeding behavior was significantly affected, but not feeding period, as shown by the top figure. At both low and high endosulfan, tadpoles fed less than in both controls. There was a significant effect for speed and a marginally significant effect for distance traveled. In both cases, there was a trend for lower travel distance and speed in both endosulfan treatments, as shown by the bottom 2 figures.
This study concluded to find that low concentrations of endosulfan in the environment altered neurotransmitters and caused locomotive and foraging differences in frogs. Endosulfan induced a significantly higher level of serotonin, reaching up to 200 micrograms, due to a direct action in the brain. The brain is rich in lipids and is therefore a target for endosulfan since it can easily dissolve into synaptic membranes, crossing the hematoencephalitic barrier to induce serotonin level changes through direct action. A serotonin increase can indicate a cell protection response against pollutants and suggest at a large range of physiological regulations such as sleeping, mood, reproduction, locomotion, and appetite. Increased amounts of serotonin in the brain decreases feeding behavior due to feedback regulation within the hypothalamus. The results of this study are in agreement with the feeding findings of other studies in that they all noted a marked increase in foraging behavior. Similar responses were observed for GABA, which was found in even higher levels than dopamine and serotonin, supporting that it is also a major inhibitory neurotransmitter in this species. The phenomenon of receptor antagonism against endosulfan could be responsible for increased GABA because animals compensate antagonistic effects by increasing quantity of ligands for that receptor. This suggests that endosulfan stimulates GABA in the presynaptic nerve terminals, inducing the release of synaptic GABA, and in turn affects the total GABA content in the brain.
In this research, there was a primary focus on the difference between the control and endosulfan treatments. There was a lack of discussion on the differences of the ethanol and endosulfan treatments. Ethanol effects need to be discussed. There was also a lack of discussion on the insignificant results the researchers were expecting. Overall, more research is needed on the neurological markers and the linkage with specific neurological disorders found in amphibians. This will aid in better understanding the complex mode of action of pollutants.